Thank you for your interest in our ultrasensitive protease sensing technology, which we use here for the detection of non-small lung cancer. We do this by means of liquid biopsies, meaning we compare the protease activity in the sera of cancer patients and age-matched, apparently healthy volunteers. This is a collaboration between Kansas State University and the University of Kansas Medical Center. Looking at the survival of non-small cell lung cancer patients, it's clearly discernible that the patient 5-year survival decreases steadily with the stage at the original cancer detection. That means the earlier we detect non-small cell lung cancer, and that's true for many other tumors as well, the higher is the anticipated patient survival. And this has profound consequences because we usually detect cancer somewhere here. If we could move this to a much earlier time, the current technology would, in many cases, be enough to really cure these patients. And that means basically that we can, let's say, save about 30 to 50% of all cancer patients just by moving the time of detection to an earlier time horizon. Our nanobiosensors for protease detection consist of an iron oxide core. We see here we have an iron core diameter about 13 to 50 nanometers, and then we have an iron oxide shell, which we use for passivation. This is then followed by an organic shell, that's dopamine, and then we attach a thread pair to the surface of the nanobiosensors, which means we have the porphyrin, TCPP, and a cyanine dye, that's usually cyanine 5.5, attached to the surface. We use about 35 TCPPs and about 50 cyanine 5.5s per nanobiosensor nanoparticle. TCPP is linked by a consensus sequence. That's a particular peptide sequence which can be cleaved by a particular protease with high sensitivity. And once TCPP is released, it starts its fluorescence. And so we can measure fluorescence intensity after incubation, which then translates to the activity of that particular protease. We see here one example. That's our findings with cathepsin B. And here we see that healthy patients have a certain activity, and stage 1 patients don't have a higher activity, but then we see characteristic increases for stages 2 and 3. And if we look at the particular box plots, then we see how these activities range. For some enzymes, we have an immediate response, and some enzymes are not conclusive. The details are all told in the paper, so I highly encourage you to go there and to look this up. What we get is a table where we have the statistical p-values for the comparison of cancer patients versus age-matched volunteers. And you see here different colors. Red means we do not have statistical significance, as we see with p-values which are significantly larger than 0.05. All the others are green, which means that we can discern different protease activity in the sera of cancer patients compared to healthy volunteers. And you see that for 1, 2, 3, 4, 5, 6 enzymes, we can differentiate between stage 1 patients and healthy volunteers. And that's exactly where we would like to see this. In fact, in our future work, we would like to move the time horizon even a little further back to really detect the transition from non-cancerous to cancerous tissue. This work would not have been possible without my dedicated co-workers, and I would like to thank my postdocs, graduate students, and undergraduates for everything they do, even in the, let's say, 10 months after the outbreak of the COVID-19 pandemic in the US. My team is still very, very spirited, and we fight for new scientific results every day. And again, I could not do it without my team, and therefore they have to be acknowledged here. We also thank the National Science Foundation for funding this study. But in any case, please spare the time and go to the original article and read, because nothing compares to reading. Thank you very much.